lecture 3 of the sequence.
This is week 3. It's kind of sort of number 2, just because the first week was an overview.
Okay.
But yeah.
I'm recording this one again. I just started the recording, so we'll just give people a couple of minutes to sign in, and we'll begin again. Thanks, Bob.
Yeah, absolutely. And I usually reserve the first 10 minutes for questions because sometimes it takes people a few minutes to get here. So we can start now. Does anybody have any questions about last week's session? Questions about the class in general? And be careful, because if you guys don't have questions, then I'm going to start asking you guys quiz questions.
Uh-oh.
And I'm going to call on people. Okay. I think next week I better come prepared with some quiz questions.
Yeah.
Let me see. I'm going to pull up something from last week.
Also, one thing I just wanted to mention, Bob, before you start, is that, we do have a Facebook PSMI, site, and, the pharmacogenomics, thread is also there as well. So if folks, want to have dialogues on the Facebook side, and it's kind of a nice way for the community to see, your questions, share questions, and if you really want your questions to be private, you can also message them privately to Bob and on the PSMI site. But I think the Facebook one might be a good way to do it too, in case people want to post questions, and will allow folks to get a little discussion going. Facebook is just when you go to Facebook, just do the @ PSMI Consulting. It's all just pretty simple. I can send an email on that as well. It's also on the PSMI website. You can just get to Facebook from there.
Who can tell me what the 4 bases are in DNA?
Ooh, is that an answer?
Who can tell me what the 4 bases are in DNA?
Hey, Bob, this is Anil Luthra.
Hi, Anil.
Hi. I missed the last session, but are you talking about GATC, GATTC? Because just from my own previous experience, is that you're referring about?
Yep, that's right. That's right.
Okay. Thank you. Well, that's
GATC.
Are you talking about guanine, adenine, cytosine, and thymidine?
That's right.
Oh, okay. Back to you, Bob.
Okay.
I thought it was thymine.
Thymine, that's right. Hey, Ron.
Hey, I'm hesitant to say anything because I don't want to teach a class. I don't want to ask any questions either.
That'll be optional.
Not that I'm qualified, really. Not.
Okay. Who can tell me how many chromosomes we have?
23.
23 pairs. I think 23 pairs.
Sorry?
I think it's 23 pairs.
23 pairs.
Most of us have 23.
23 pairs, right?
Mm-hmm.
Okay. Okay.
Now, who can tell me that they're on Google?
I'm not.
Oh no!
Me.
I actually have my 23andMe profile up on the screen. Okay.
Wow.
So within
I don't actually have it on the screen, but I have
so
done.
within the within the cell, is there DNA anywhere other than in the nucleus?
I'd say no.
Say no? I'm going to ask you again after today's session.
Dang, wrong answer.
Huh? I'm not saying anything other than I'm going to ask you again after today's session.
Are you
any
counting
other,
RNA too, or just DNA?
Just DNA. Is there any other DNA in the cell other than what's in the nucleus?
Mitochondrial DNA.
Mitochondrial DNA. Okay.
I thought that was RNA.
Okay, so we'll cover that today. Yeah, I have to torture you guys with questions. The first 10 minutes is going to be for questions. And if you guys don't have questions, I'm going to ask you guys questions from the previous week. But I think I've about run out of questions. So if you guys don't have any more questions, we'll go ahead and get started. Anybody have any further questions before we dive in this week? All right, good. So, thank you everybody for sending back your responses to those questions from the first overview, where some of the questions asked about your baseline knowledge of genetics. And based on that, I think what we had planned on covering in 3 weeks, we can cover in 2 weeks. So today we had planned on covering just the overview of cell and cell division. Then next week we were going to do the central dogma biology. And then on the 25th of May, we were going to do genetic mutations and polymorphisms. But we've modified the curriculum a little bit so that these 3 sessions are going to be covered now in 2 weeks. Because it seems like most people, in fact, from the responses I got, everybody kind of sort of understands the basics. So the curriculum's been a little bit altered. We'll shave off a week, so now we'll finish on July 13th instead of the 20th. And again, this is kind of a dry run, and so there might be further changes as we go. But I'm still keeping the slide decks topic specific. So that means today we're going to go through this slide deck, through slide deck number 2 in completion, and we'll go halfway through slide deck number 3. And then next week we'll finish slide deck number 3, and then we'll do all of slide deck number 4. And it also means that we've only posted so far slide deck number 2 on the website. Slide deck number 3 will be posted next week when we get to the tail end of it. So most of the slides from today that's in this deck you'll see on the website, and then slide deck for topic number 3 we'll post next week. So we're going to start every class with 10 minutes of questions. Some of this material is old hat to a lot of you. Jonathan? Someone's saying hello out there. So some of this material is old hat, and some of this material will be new. And particularly since we've kind of decided to accelerate the content a little bit, some of it may leave you feeling like you have some questions. So do feel free to bring questions to class. If you are in any way, shape, or form embarrassed to do that, you can always feel free to send your questions directly to me or to Jim. And we'll make sure to, and of course, you're always welcome to send us feedback, positive or negative, about the class. So today, as with most classes, we're going to start off with a clinical case. This is a case of myoclonic epilepsy with ragged red fibers. Admittedly, a pretty rare condition, the one that illustrates some just absolutely fascinating genetic principles. And of course, what we're going to do every week going forward is for every clinical case, we're going to carry it through not only the clinical case and the clinical genetics aspects of the case, but we're going to then explore the bioinformatics and the clinical informatics aspects of the case. So what we're going to try to do for each clinical case is carry it all the way through from clinical to clinical genetics to bioinformatics to clinical informatics, so that you can understand the disease, understand the genetics of the disease, but also understand how to poke around and learn more about the genetics of that disease through various bioinformatics portals. And then also understand from an HL7, SNOMED, LOINC perspective, how do you actually communicate information about that disease from one person to the other. And what we're going to do is kind of sort of start off the bioinformatics and the informatics discussions with relatively simple scenarios, but then the bioinformatics and the clinical informatics scenarios will get increasingly more interesting, possibly a little bit more detailed, but certainly more interesting as we go forward. And then today we're going to talk about the cell and cell division in this deck, and then we'll switch over to deck number 3 to finish out the day with a discussion of the central dogma of biology. Okay? So before we start the case, I wanted to give you a couple slides on pre-case review. So 2 slides on pre-case review. Last week we talked about the fact that the cell has a nucleus and that the nucleus contains 46 chromosomes, each comprised of DNA. Today we're also going to be exploring mitochondrial DNA. Cells have a variable number of mitochondria, also sometimes affectionately referred to as the powerhouse of the cell. And each mitochondrion has a variable number of DNA molecules in it, somewhere around 10 DNA molecules per mitochondrion. So we're going to talk about mitochondrial DNA today and its inheritance properties and its other properties. The other pre-case review thing I want to talk about is the fact that 50 years ago, when we thought about genetics and we thought about the central dogma of biology, really the only model we understood was that a chromosome has a gene. Genes are transcribed into messenger RNA. Messenger RNA is translated into protein. So that was kind of our worldview of genetics 50 years ago, but we've learned obviously quite a bit since then, and we now know that there are probably at least, if not many more, non-protein coding genes. These are stretches of DNA that are transcribed into RNA, but that RNA does not direct the cell to construct proteins. So historically we've been taught about protein coding genes, but now what I'm telling you is that we have at least as many, if not vastly more, non-protein coding genes that simply get transcribed into RNA, and that RNA is not translated into protein, but rather that RNA serves one of a myriad of functions, some regulatory functions, some cell structural functions, a number of functions. And we'll be talking specifically in this case today about a mutation in a non-protein coding gene, so that you begin to have a understanding of how mutations in your DNA outside of the protein coding genes can also result in clinical manifestations, and also how do the standards like SNOMED, like LOINC, like HL7, how do those standards accommodate communicating mutations in non-protein coding genes? And feel free to stop me along the way if you have questions. And I'll do my best to answer you. Sometimes, honestly, as much as I love this stuff and as much as I've been reading, I feel like the scope of genetics is so broad that it's almost intimidating to me to try to teach this material to you guys. It would be easy for you guys to stump me with questions. But I know we also have on the call some experts in various aspects of genetics that can help if there are questions. And of course, any questions that I can't answer, I'll write down and get back to you next week on that. So we're going to talk today about...
Hey, Bob, I do have a question. Bob,
yeah, Ron?
On your last slide, you know, there's this term that's bandied about called junk DNA, which I think is a misnomer. We just don't understand it well enough. But is the non-protein coding gene synonymous with junk DNA?
And next week we're going to be specifically looking at junk DNA. And I'll tell you, this is kind of sort of. We used to think that everything that wasn't a protein coding gene was so-called junk DNA. We now believe that as much as 80% of our DNA serves some functional role. Okay, and some of that 80% is non-protein coding genes, but even this other portion of the DNA serves functional roles. They serve, for instance, as binding sites for enhancers. And so we used to think that, you know, this was all junk. We now believe that as much as 80% of the DNA serves some functional role, a portion of which are these non-protein coding genes. But the junk DNA would also have applied to other stuff.
Thanks.
Okay. Mm-hmm. So this is a 15-year-old male who was referred, by the way, this is a muscle biopsy of this 15-year-old male who was referred to the neurogenetics clinic for epilepsy. His EEG was consistent with a seizure disorder. He had been well, developing normally. In his family history, he had his maternal uncle, okay, so his mom's brother, died of some undiagnosed type of muscle disorder at the age of 53. His maternal aunt had progressive dementia and developed ataxia, or a funny abnormal walk, at the age of 37. And an 80-year-old maternal grandmother was deaf, had diabetes, and kidney disease. This particular patient had generalized muscle wasting and weakness. He had myoclonus. He had an abnormal gait called ataxia. And when he was evaluated, it was found that he had sensory neural hearing loss. He was going deaf at the age of 15. He had slowed nerve conduction velocities. His blood and his cerebrospinal fluid lactate levels were elevated. So they did a muscle biopsy on this guy. And what they found here was a couple of things. They found these ragged red fibers. So if you can imagine, this is a muscle cut cross-section, and you can see these are these long fibers that were just cut. But some of these fibers look funny. They stain positive for mitochondria right underneath the sarcolemma, or the plasma membrane of the cell. And over here on the right, what they've done is they've stained these muscle cells with a special stain looking for a chemical called cytochrome oxidase. And what you see here, well, actually what you don't see here, is any positive staining. These cells don't stain positive, and particularly this one has almost no cytochrome oxidase in it. So something is very abnormal. Cytochrome oxidase is a protein enzyme produced by the mitochondria. And so we see that the mitochondria look abnormal in this muscle fiber, and we see that these cells are staining negative for cytochrome oxidase, which is a protein produced by the mitochondria. Based on that, his team presumed that he had a mitochondrial disorder and went ahead and tested him for a specific mutation associated with this condition, myoclonic epilepsy with ragged red fibers, or we can just call it MRF. And in fact, found that he did have this mutation. They went ahead and they tested his mother, his aunt, his grandmother, and in fact, they tested autopsy material from the deceased uncle and also found that they, in fact, had the same mutation. So here's the patient that is found to have a mutation in his mitochondrial DNA. They found that same mutation in mom, in maternal uncle, in maternal aunt, and maternal grandmother. And no other family members agreed to be tested for this condition. So this is a case of myoclonic epilepsy with ragged red fibers due to inheritable mutation in mitochondrial DNA. So we're going to talk a little bit about mitochondrial DNA. And I'll tell you, this is a pretty rare disease. And generally, I try to include more common diseases. But mitochondrial diseases overall are pretty uncommon, but they're just so darn fascinating because oftentimes it's through disease that we come to understand, the DNA and how the DNA works, because if somebody has a mutation, it helps us understand what the role of that segment of DNA was. So, here we're going to talk about a rare disease only because it's just so darn fascinating. Mitochondrial DNA, it's a circle of DNA. It's 16,569 base pairs and it contains 37 genes. There's about 10 of these circles in every mitochondrion, and a given cell can have 10 to hundreds of mitochondria, depending on how metabolically active that cell is. Highly energy requiring cells will have a whole boatload of mitochondria, and each mitochondrion will have roughly 10 of these segments of DNA. This DNA codes for 37 genes. Most of these genes play a significant role in energy production, or what's called oxidative phosphorylation. And you can see here in green the cytochrome oxidase enzyme, which is actually three separate genes. The protein products of these three genes join together to form the cytochrome oxidase. And this was the protein that was missing in the muscle biopsy. Although, as we're going to see, the specific mutation isn't in this particular gene. It's in a separate gene that produces RNA that actually doesn't get translated into protein, but rather, in this particular case, the mutation, which is in the gene right here, codes for an RNA, and that RNA plays a critical role in enabling these genes to fully produce their protein product. But before we get into that, there's some really fascinating things about mitochondrial DNA. It doesn't work the same as nuclear DNA. So we're going to talk a little bit about mitochondrial DNA, replication, and inheritance. And these four topics in particular we're going to cover because they differ from nuclear DNA. The first is maternal inheritance. So what happens is the sperm loses all of its mitochondria, whereas the egg retains its mitochondria. So in the fertilized egg, the sperm that enters the egg either contains no mitochondria whatsoever, or if there's any mitochondria left, those mitochondria get destroyed when the egg is fertilized. So virtually all inheritance of mitochondrial DNA is maternal. And you'll see that in these family trees or these pedigrees where if a male is affected, then 0% of his offspring will inherit the disease because there's no way for the male to pass on his mitochondrial DNA. Whereas if the female has it, she will transmit that mutation to 100% of her offspring. So this is a classic pedigree demonstrating maternal inheritance. And if you see a pedigree like this, it would clue you in that this is a mitochondrial DNA disorder where 0% of the male offspring have the disease and 100% of the female have the disease. And again, this is because the maternal mitochondria get into the egg, whereas the paternal mitochondria don't survive in the sperm. The other thing that's very interesting about this mitochondrial DNA is the number of affected chromosomes, the number of affected mitochondrial DNA strands will vary from cell to cell for two reasons. One is illustrated on this slide and is referred to as the oocyte bottleneck, and the next is what we're going to talk about on the next slide, called replicative segregation. When the egg is being formed, initially the primordial germ cell has a whole boatload of mitochondria. And you can see that the red ones have the mutated DNA and the blue ones don't. But during the process of egg formation, the number of mitochondria are drastically reduced. And so here you see, as the oocyte is maturing, you see various primary oocytes having had the mitochondria reduced. And just by chance alone, some of these primary oocytes are going to have mainly diseased mutant mitochondria, whereas some of these oocytes are going to have almost normal mitochondria. And as those oocytes go on to mature, you're going to wind up with some oocytes that are highly abnormal and some that are almost essentially normal. So because of the way the mitochondrial DNA replicates, and because it's not uniform across all the mitochondria, during the process of oocyte maturation, you'll wind up with eggs that vary considerably in the amount of diseased mitochondria. Also, see, when the cell divides, we haven't talked about mitosis yet, but I think most people understand that when the cell divides, you have 23 pairs of chromosomes in the nucleus. And when the cell divides, there's an orderly segregation of those pairs. You know, chromosome 7, the one you got from mom goes to the left, and the one you got from dad goes to the right. And chromosome 9, you know, the one you got from mom goes one way, and the one you got from dad goes both ways. You know, it would be abnormal for, both chromosome 9s to go into one daughter cell and none of the chromosome 9s go into the other daughter cell. So the segregation of nuclear DNA during mitosis is very orderly, very well orchestrated, in contrast to the segregation of mitochondrial DNA. The mitochondria go through a process of fission. It's loosely coupled to the cell cycle in that the mitochondria begin to divide themselves prior to mitosis, but then the divvying up of the mitochondria is a random process. It's not an orderly process. And that means, as a result, some daughter cells can receive a considerable number of mutant mitochondrial DNA molecules, whereas other daughter cells might receive almost no mutant mitochondrial DNA molecules. And that means that when a cell divides, the daughter cells can actually differ in their phenotype. Some of the daughter cells can be more diseased than other daughter cells. And this process is called replicative segregation, and it's really one of the things that makes mitochondrial DNA diseases so tricky is that as a result of this replicative segregation, the symptoms can vary enormously amongst patients, and they can vary enormously with respect to severity. They can vary enormously with respect to, in this person, the brain, the CNS effects might be very prominent, whereas in this other person, the hearing loss might be more prominent, and in this other person, the skeletal muscle manifestations might be more prominent. You wind up with a condition in a cell known as homoplasmy and heteroplasmy. In genetics, a lot of the vocabulary that we're going to encounter, at least when I poke around, these definitions aren't completely stable across sources. And so when you see a word homoplasmy, some sources say that homoplasmy means that all of the mitochondrial DNA within a cell is the same, whereas heteroplasmy refers to a condition where within a cell you have different forms. You have some normal mitochondrial DNA and you have some mutated mitochondrial DNA. Other people have used these words slightly differently because, even within an individual mitochondrion, some of those DNA strands can be normal and some of those DNA strands can be mutated. So sometimes you'll see homoplasmy and heteroplasmy referring to the cell as a whole, but sometimes you'll also see these words being referred to individual mitochondria. Is the individual mitochondrion homoplastic or heteroplasmic? But homoplasmy and heteroplasmy is the rule for mitochondrial DNA, and it's because of the heteroplasmic nature that also contributes to the tremendous variability in severity of disease manifestation for these mitochondrial DNA disorders.
Bob?
Yeah.
Two things. So what you're saying now is that earlier slide showing 100% inheritance of from any maternal side actually wouldn't be completely true. It might be that on some random basis there'd be a rare offspring that would not have mitochondrial disease. Is that correct?
Yeah, that's exactly right. And in addition to that, you'd expect that so you might see zero symptoms in some of these people just because of the random nature, but
Right.
you also might see extreme variability in the symptoms in these
Right.
other patients.
Okay.
That's
And
right.
I don't know if you're going to get into it, but there now is a treatment for this. It's kind of the three-parent treatment. I don't know if you are going to talk about that.
Oh no, darn it! I will. Yeah, let's talk about, because in fact I mentioned there's no gene therapy, but you're right, there is gene therapy, I guess, in a way, isn't there? Yeah.
Yeah.
Okay,
you
we'll
out
come.
the mother oversight
Yeah.
and somebody else's. Yeah. Go ahead. I'll let you.
Okay, we'll come to treatment. Yeah, we'll cover that. We'll talk about that. Thank you. So this is another picture of the mitochondrial DNA. And this gene right here is what gets mutated in MERF. And you have an A at position 8344 is changed to a G. And this particular gene produces an RNA. That RNA does not get translated into a protein. Rather, this RNA plays a critical role in the ultimate protein translation from the cytochrome oxidase 1 cytochrome oxidase 2 and cytochrome oxidase 3 components. So basically, even though you haven't mutated the cytochrome oxidase gene, because you have a mutation in the transfer RNA gene here, the levels of cytochrome oxidase are drastically reduced. The mitochondria simply can't produce cytochrome oxidase because they're missing a key enabler, and that's the RNA produced by this particular gene. And that's an important thing to understand because, again, last week we talked about sickle cell disease. And sickle cell disease is great on the one hand as a tool to illustrate the flow from genotype to phenotype because genotype and phenotype are so tightly coupled. But that's not necessarily real world. What we see in general is that for a given phenotype or for a given disease, mutations in a number of genes can result in the same or very similar phenotype. And that kind of sort of is illustrated here, where if you had a mutation directly in the cytochrome oxidase gene, that would give you muscle disease, but having a mutation here in this RNA gene acts as if you had a mutation in the cytochrome oxidase gene. They result in similar or at least overlapping phenotypes. And that's generally the case in genetics, is that mutations in any number of genes can result in a similar phenotype. That's actually referred to as locus heterogeneity. And let me just remind you guys that these slides are mainly pictures. There's not a lot of text. But if you do download the slides, you'll see there's text associated with some of the slides down here in the notes section. But I don't generally put the text on the slide because I just prefer the pictures. So if you want some of the text, just feel free to grab the slides off the website. This slide here just kind of shows there's a lot of other known mitochondrial DNA diseases. Now, most of these diseases can also, again, just like we talked about, be caused by mutations in the nuclear DNA. So, for instance, diabetes mellitus. While you can get diabetes from certain mutations in the mitochondrial DNA, 99% of diabetes has nothing whatsoever to do with mutations in mitochondrial DNA. So just like with most of these, while you can get these manifestations from mutations in mitochondrial DNA, most of the time that's not actually the culprit for a lot of these phenotypes. And so here, Jonathan corrected me. I was going to talk about treatments because Jim wanted to know about treatments, and then I completely blanked out, and I'm going to have to fix this slide. So some people have tried coenzyme Q10 and L-carnitine to increase mitochondrial energy production. The jury's a little bit still out, even though there's been some very interesting success stories. But the main type of genetic engineering treatment that actually is being used, in fact, it's been approved now for use in the United Kingdom, is the three-parent solution, where what they'll actually do is they'll take, so mom and dad are married, okay, but mom has the mitochondrial DNA problem, and dad is normal. What they'll do is they'll get a donor egg from some other female. They'll get a donor egg from some other female that has normal mitochondria. They'll take mom's nucleus from her ovocyte, and they'll implant mom's nucleus into the other female's egg. They'll remove the other female's nucleus from that egg. And, gosh, I should have a picture, but is that kind of sort of clear? What they're literally doing is they're getting a donor, they're getting her egg, they're removing the nucleus from that egg, They then take mom's egg that has the mitochondrial DNA defect. They're taking her nucleus and they're transplanting her nucleus into the oocyte of the donor. They then fertilize that egg with dad's sperm. And so what you wind up with is you have an egg that has the mitochondria from the donor, the nucleus from mom, and then that's fertilized with the sperm of dad. And that then could be a cure for the disease. And they're actually now allowed to do that in the United Kingdom. I don't think that's quite yet been allowed in the United States, but if not, probably soon. Jonathan, do you know if that's allowed now in the U.S.?
I think there was controversy about this in the paper just a couple months ago, and I think it did get approval here after, it was approved in England first and was not approved here, but I think it did come through that we got approval.
Okay.
It's like a transplant medicine, except we're at a whole different scale, huh?
Yeah, I probably can find a quick picture here. Let me find. Anyhow, you kind of sort of get the message. I don't want to get too into this, but it's so you have mom's egg has faulty mitochondria. They're replacing the donor egg with mom's nucleus, and then they do in vitro fertilization. And lo and behold, you have the perfect baby. The jury is still a little bit out on this. One of the reasons is because there are known interactions between nuclear genes and mitochondrial genes. Mitochondrial genes play a role in energy production, and nuclear genes also play a role in energy production. And so it seems as though the kids that have been born with this triple approach are just fine. But, as always, people urge caution, and they say more studies are necessary and longer follow-up periods are necessary before you can be completely comfortable with it working. Is that what they call the
three-parent babies?
That's right. Yeah, the three-parent babies. That's what they are. Okay. So, gosh, I can't believe I screwed up the slide. That's why it's a dry run. So let's take a look at the bioinformatics aspects. And again, I distinguish between bioinformatics and informatics, where bioinformatics is the stuff that geneticists do. It's all of the tools that are out there now to explore genetics and variants, whereas informatics is kind of sort of what we do. It's, how do you interface this stuff with electronic health records? And they're both super fascinating fields, and as you can imagine, both pretty huge fields given the explosion of genetics. So what I thought we would do for this case would be, so the case of MERF, what we saw was at position A344 on the mitochondrial DNA, an A was substituted with a G. So let's look specifically at this mutation. And first, let's just look at it, the mutation, and then let's track through some of the bioinformatics portals to kind of see what we can learn about this variant. The first thing is we can explore this variant. Now, the gene that's mutated is the MTTK gene, okay? So if we go, and by the way, you can click on this, and it takes you to the UC Santa Cruz genome browser, okay? Now, if you click this link, you're not going to see exactly what I'm seeing here. The reason is because you can configure this genome browser to show various what are called tracks. I have it configured to show the genes And I have it also configured to show simple point mutations. But all of these are various tracks that you can also decide to see. Let's say, for instance, I want to show a couple other tracks, just to give you an idea. I got to find some bigger tracks. So there's a bunch of stuff. So here's the gene, still here in green, but there's a lot of different tracks, or what are called annotations, that you can choose to see or not. And depending on what tracks you've enabled, when you click the hyperlink, it's going to show you this stretch of DNA, but it's going to show you the tracks that you picked to show. Oh, well, this is a long process, isn't it? And we'll actually look at some of these other tracks. Oh, okay. Well, I'm going to quit erasing those. Here's the gene. What you see when you're looking at this, first off, is we're looking at USC Gene Browser. UC Santa Cruz Genome Browser is probably the most popular gene browser worldwide. And we're looking at reference chromosome 37, or HG19. And what the GRCH37 stands for is the Genome Reference Consortium, Human Genome Build 37. Okay, so the Human Reference Genome, they have Build 37. This came out in February 2009. And then UC Santa Cruz takes that build, and then they give it an identifier of HG19, and then they build all of their annotations off of this reference chromosome. Obviously, everyone's DNA is different, and so the NIH develops every 5 years or so a complete reference genome. There was a reference genome. This would have been GRCH37. Actually, there's a new one, 38, or HG38, which came out in 2013. But the thing is, so many annotations have been applied to this particular build that it's still extremely popular and useful to reference. So at the top, you'll see what reference you're looking at. You can zoom in, you can zoom out, you can zoom to specific areas. This is a relatively small piece of DNA, right? It's only about 17,000 base pairs. So this is the entire stretch of mitochondrial DNA. You'll see it abbreviated as chromosome M, or you'll also see it abbreviated as MT DNA. And the little red box shows you where we've zoomed in on. You can see our magnification here. This is only 50 bases. And we're specifically interested in the mutation at position A344. Position A344 is relative to the complete chromosome. So going from position 1 to position 16,569, when you get to position 8,344, which is right here, then the mutation we're specifically interested in is where an A has been substituted with a G. And when you see right down here, there's a single nucleotide polymorphism that's been identified at that spot. And that's this identifier you see in the slide deck, RS118192098. So we're specifically interested in this mutation, which is a change at position 8344 from an A to a G. And that's a known mutation, and so it's been identified and it's been logged in the SNP database, single nucleotide polymorphism database. And what you can do is, if you're interested in learning more about this particular variant, you can click there. And if you click on this link, this is the dbSNP identifier. If you click on this link, it takes you to the NIH dbSNP database. Okay? So now we're in the dbSNP database, which is a database of short variations. Now, unfortunately, there's nothing out there on the internet that is 100% complete with respect to all known variants in human DNA. The NIH databases, are very good. They're very thorough, but and I'll show you as we go forward. Sometimes you're simply not going to find what you're looking for in these databases, and there's other techniques you can use if what you're trying to do is explore. Your lab reports back your patient had this particular variant. Okay, well, oftentimes you're going to come look in dbSNP and learn more about it. Actually, what's typically going to happen is the lab is going to report back what that variant is. But if you want to poke around and explore a little bit more, oftentimes you're going to find information about that variant in the dbSNP database. The dbSNP database is a catalog of what are referred to as small variations. And small is kind of relative, but typically small variations mean under 50 bases are involved. When the variation is larger, when more than 50 bases are involved, there's a separate database called dbVar, and we'll look at dbVar for a different case in a different session. So dbSNP is a catalog of small variations, usually under 50, whereas dbVar is a catalog of large variations, typically over 50. And there's a lot of reasons for having to separate these. The other thing about dbSNP is the way that they handle identifiers. So let's say, for instance, here we have at this particular position 8344, an A was mutated to a G. Let's say instead at position 8344, an A was mutated to a C. From dbSNP database perspective, the identifier would be the same because this identifier is unique to a specific position on a reference chromosome and a specific length. So any point mutation at this position will share the common dbSNP ID, regardless of what that specific mutation is. So whether it's an A to G, whether it's an A to C, if it's a single point mutation at this particular point, it's going to get the same dbSNP identifier. In this particular case, when we see this particular variant, this variant happens to be identified as pathogenic, and there happens to also be a reference to a description of this variant in the ClinVar database. And the ClinVar database is kind of sort of a subset of the dbSNP database, where those SNPs that are known to be clinically significant are cataloged. And I say kind of sort of because there are entries in ClinVar that aren't necessarily in dbSNP, because somebody may have submitted something to ClinVar without submitting something to dbSNP. So you can kind of sort of think of ClinVar as being the best place to go if you want to look up the clinical significance of a point mutation. And you can kind of sort of think of dbSNP as the place to go if you just want to explore everything you can about a point mutation that may or may not be clinically significant. But later in the session, we're going to look at some exceptions and how you can kind of poke around the internet and find out more information, even if the mutation you're interested in doesn't happen to be in ClinVar and doesn't happen to be in dbSNP. But in this case, this particular mutation is in dbSNP, and in dbSNP it's thought to be pathogenic, and there's a corresponding link to ClinVar. And the other thing you see here is the HGVS notation. And the HBGS notation is very useful because this is the key that you'd use to search ClinVar. You might know the ClinVar ID, or you might know the SNP ID and can link to it from here. But if you wanted to search ClinVar, the other way to search ClinVar for a particular mutation is to search by way of its HGVS string. And in this particular case, HGVS has a reference sequence, okay, NC012920.1, and does this show the reference sequence ID? No, okay. This is the reference sequence that we're talking about here, the mitochondrial DNA from GRCH37. Okay, and then this is when they count, position A344 again is 8,344 bases from the start of the mitochondrial DNA, and then at position A344 and ASF2 to a G. So we could search ClinVar for this HGVS string, or in this case, we can simply click it, and we'll go to ClinVar. And once we go to ClinVar, we'll see a couple of interesting things. First off, this is the ClinVar ID, and we'll need the ClinVar ID when we generate our HL7 messages, because the ClinVar ID, that's the primary identifier for the variant in an HL7 message. Sometimes you can use other identifiers in the HL7 message. For instance, you can use the dbSNP identifier, but they're not as good because, again, the dbSNP identifier is specific to a location, but it's not specific to the actual mutation. So the ClinVar identifier is the single best identifier for a variation to use in an HL7 message if you have one. But a couple of the other things you'll notice here is this is getting back to a question that Jim asked last week. Is this a pathogenic mutation or not? And in ClinVar, what you see here is that there's conflicting interpretations. And most people presume that this is pathogenic, but one submitter said, well, it's possibly pathogenic, but I'm not sure. And so ClinVar sometimes will show you conflicting information about the pathogenicity. And the other thing that ClinVar will show you is that this particular mutation has been seen not just in MRF, but people have also reported seeing this mutation in Leigh syndrome and in a fairly rare type of Parkinson's disease called mitochondrial Parkinson's disease. And so we said mutations in different genes can result in the same phenotype, but it's also the case that a single mutation can be responsible for multiple phenotypes. So you kind of have this many-to-many relationship between diseases and phenotypes, okay? And then the question is, now that we kind of know a little bit more about this particular variant, and we believe clinically this person has myoclonus with epilepsy, now maybe we want to know what the SNOMED code is. So through OMIM, you can't get directly to the SNOMED code through MedGen or OrphaNet, but if you go to OMIM, you'll find when you look up a disease in OMIM, you'll usually have the codes up here. And in this case, it gives you a choice of 3 different SNOMED codes. And, possibly this is an indication that SNOMED needs to be cleaned up a little bit. But this stuff is complicated. And, I think this SNOMED code is right. I think this SNOMED code needs to be retired. And I think this SNOMED code, some people believe this is actually for Fukuhara disease. And you can see Fukuhara was one of the guys who first reported MRF. And so, possibly Fukuhara disease is simply a synonym for MRF. But it's all very complicated because back when Fukuhara reported the disease, MRF maybe wasn't described, and so he got a SNOMED code for Fukuhara disease, but then MRF was better described. So the SNOMED coding for these diseases can be very complicated. So we've determined from OMIM that there's 3 possible SNOMED codes, and now we want to communicate the information. This is a simple mutation as opposed to a complex or structural mutation. We looked at a simple mutation last week. We looked at the HL7 version 2 in the FIRE instances, and simple mutations are all communicated kind of in the same way. So let's look again at the V2 in the FIRE instance. In a V2 message, you're simply going to have a panel where you're going to have a number of LOINC codes, and then you're going to have answers. And in this particular case, this is a simple variant, and we have the ClinVar ID 9579, and the display name for that ClinVar ID is the HGVS expression. The HGVS expression has the reference sequence with the gene in parentheses, followed by the actual mutation. Having communicated the ClinVar ID, some of this other information is redundant, but you can send the actual gene. Here's the code for the gene, but most people think of genes by their display names, the gene symbols. The actual transcript reference, the actual DNA change. Here, where you see that the clinical significance is pathogenic and the phenotype is this particular SNOMED code, what's happening here is that when a lab reports this mutation, the lab will have decided what they want to report back as pathogenic and what they want to report back as the associated phenotype. Now, 2 labs potentially will report back the same variant different, because you'll see that there's conflicting evidence. But typically, a lab will report back, if the preponderance of evidence is that it's pathogenic, they'll report back pathogenic. If they're not sure, they'll report back some other value here, such as likely pathogenic, or pathogenicity unknown, or we have no friggin' idea what the pathogenicity is. And there's millions of variants out there for which the pathogenicity, the clinical significance, is unknown, and that'll be something we'll be talking about in a few weeks as well. What the heck do you do when you find a variant and you have no idea what its pathogenicity is? But in this particular case, the lab has chosen to say that this mutation is pathogenic, that it's associated with this SNOMED code. And what they do here is they determine whether or not the mutation was heteroplasmic or homoplasmic. This will become clearer after we talk about next generation sequencing. But basically what's going to happen, for instance, is during next generation sequencing of the mitochondrial DNA, you're going to get, at this particular position, you're going to have some reads are going to show an A at this position, and some reads are going to show a G at this position. So therefore, they know that there's a blend of mitochondrial DNA, and therefore infer that this is a heteroplasmic mutation. The fire example.
So, Bob, that's a beautiful. That's a beautiful message. Is that a hypothetical message, or was that from an actual laboratory transmission? Because in the real world, I just so often just see some horrible blob of text followed by a comment from the medical director that this might mean that the patient has such and such a disorder.
Right. Well, this is fake. So I made up this example, but I made it up based on the Dolin codes in the September 2016 ballot of the genetic test result message. So I asked him in my ballot comment to include a mitochondrial DNA example, but this one I just made up.
I know it's beautiful. I wish that we actually could see this in the way that laboratories are recorded in real life.
Yeah, I agree. The fire example is kind of big. What I did was, so the fire examples are pretty big, and what I did was I put these fire examples, they're also on the website, so they're a little bit too long to go through right now. They're equivalent in semantics, and if you want to take a look at the fire instances in more detail, just feel free to grab them. Feel free to grab them off the website. Okay. Okay. So let's see. That took a little bit longer to explain than I thought, but I can't help it. It's just very interesting. We're probably not going to talk much more about mitochondrial DNA through the rest of the course, again, because it's so rare, but I just had to do you guys justice by introducing you to mitochondrial DNA. So we're going to switch focus now, and I think when we talk about the cell and cell division, I'm going to probably try to speed it up a little bit. But the remainder of the material, the cell division, and the central dogma are, I think most of you guys already have a pretty good understanding of, so I'm going to probably now go a little bit quicker through the rest of the material. This slide mainly is just to give you an idea of the size of a cell and how you need a light microscope really to see it. We're kind of talking about this range right here. And probably most of you are familiar with the cell. And here we have the nucleus, and here's all the DNA. And in particular, we have these organelles, the mitochondria, which is the energy producer. We have, I hear some background noise.
Yeah.
Thank you. And here we have, actually, some of these organelles are important for protein synthesis, particularly these little red dots, which are called ribosomes. This network here, called the endoplasmic reticulum, which here is just studded with these ribosomes. And then we have these Golgi vesicles over here. And I thought it might be helpful to zoom in on some of the cellular components that are involved in protein synthesis. I guess partly just because this stuff just boggles my mind to think about how all of this works. But the DNA is inside the nucleus. The RNA transcription comes out, and the ribosomes here then read the RNA and produce protein within these within the endoplasmic reticulum. Pieces of the endoplasmic reticulum then bud off and form the Golgi apparatus, and within the Golgi apparatus, the proteins are further processed and some of them are then taken to the cell surface where they're excreted, or they may be released into the cell as little vesicles. And so it's this really tremendous mechanism of where the DNA is directing protein synthesis, and then those proteins traverse their way through the cellular machinery to their ultimate destination, wherever that might be. Within the nucleus, it's not just a plate of spaghetti. It turns out that each chromosome occupies a specific territory, and it's not simply random, for instance, that this particular chromosome is contiguous with this particular chromosome. There's dynamic interactions that are taking place between the loops of the chromosome. And in fact, even the loops of the chromosome play a functional role because the looping can expose or can hide segments of DNA to enzymes responsible for transcription. So the organization of the chromosomes within the nucleus is highly ordered and highly functional, and we probably have barely even scratched the surface of understanding what the three-dimensional layout of the chromosomes in the nucleus, what the significance of that is in terms of regulation of gene production. And we'll talk more about that. Next week we're going to talk about what's called epigenetics, or how does the cell decide which cell is going to produce the protein product from which particular gene, and exactly when is that going to happen. And that whole process, which we refer to as epigenetics, is in part based on the three-dimensional structure of the chromosomes. And we'll come back to that in more detail next week. The cells go through the cell cycle, and while we're mainly talking about cell and cell division here, I want to dwell a little bit on the cell cycle and introduce you to it because it's going to become critically relevant when we talk about cancer genomics, because there's hundreds of protein products that govern the traversal of the cell through the cell cycle. Hundreds of protein products. And as it turns out, many of those genes that code for the protein products that govern the cell cycle, many of those genes, when mutated, result in cancer. And so understanding the cell cycle is kind of like hand in hand with understanding cancer genetics, because so many of the mutations responsible for cancer are mutations in genes that code for proteins that play critical roles in the cell cycle. So cells typically spend most of their life living in this G1 phase. This is just when the cell is doing its thing. It's, it's filtering the blood. It's, whatever, pumping. It's, it's doing whatever it needs to do. Some cells never divide. They are just in this phase forever. Sometimes that's referred to as G0 stage. But at some point, the cell decides it needs to divide. And during the G1 phase, the cell ramps up. It gathers together the nutrients that it's going to need to divide. It, the mitochondria go through a process of fission. Everything doubles up, or everything gets ready to double up. And then when you get into the S phase, or the synthesis phase, the chromosomes replicate. So you have a chromosome, and it now replicates. And now you kind of sort of have two chromosomes glued together, and you call these chromatids. So now you have a chromosome that has chromatids, and then you go through this period where we make sure that everything is ready to go, that everything's all set, and then boom, we have mitosis, and the cell splits, and we now have two daughter cells, and then that daughter cell, possibly may continue to go through the cell cycle, or it may rest. And along the way of the cell cycle, we have various checkpoints, and these checkpoints also play a very important role that we'll come back to when we talk about cancer genetics, because oftentimes the proteins that play a role in checkpoints are mutated. The genes that code for those proteins are mutated. So we might have a checkpoint here before the cell from into synthesis that makes sure that the cell is big enough, that it's strong enough, that it has all of the chemicals that are necessary for it to divide. And then, okay, the checkpoint says all good to go. Now we let the cell go through the DNA synthesis phase, but still before going through mitosis, there's another checkpoint that makes sure that the DNA is undamaged, because if the DNA was damaged during replication, the cell might need to auto-destruct. The stuff, MPF, and again, if you look at the bottom of these slides, there's all of the text that kind of describes what all of this stuff means. And I don't really want to talk about MPF or maturation promoting factor now. The only reason I mention it is so that when we talk about it during the cancer genetics session, you will have heard it once, and so it'll be that much easier to kind of understand what's going on in cancer. The maturation promoting factor has these proteins, cyclin-dependent kinase and cyclin, and it promotes the entrance of the cell into mitosis by a process of phosphorylation. It activates a lot of the enzymes necessary for going through mitosis. And sometimes in cancer, the MPF proteins are out of control, and they start phosphorylating proteins even when they haven't been told to do so. And then the cell just says, okay, cool, let's go. Zip, 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 zip. And the cell just keeps cycling out of control because MPF proteins were mutated, and therefore the brakes are off. There's no checkpoint there anymore. So cell cycle is important, critically important for an understanding of cancer genetics. And then ultimately, we reach a point where the cell is ready, and it goes through a process called mitosis. And mitosis is a process that I think most of you are familiar with. Oops, here we go. Mitosis, where the cell has made it through the synthesis phase, right? All of the DNA has been copied, and the cell is nice and big and plump. The organelles have been doubled up, and now we go through mitosis. And during mitosis, you have 46 chromosomes, and all 46 of those chromosomes line up. You have all 46 of those chromosomes lining up, and this chromosome splits, and half of that replicated chromosome, these two chromatids, go to the left, and half of that goes to the right. So for each of these 46 chromosomes, you've got half going to the left and half going to the right. The cell pinches off through a process called cytokinesis. The cells divide. The nucleus reforms, the nuclear membrane reforms. And there you have two new cells. And of course, this is the normal way that DNA lives, is decondensed chromatin. The DNA, the chromosomes only condense during mitosis, but normally the DNA lives as these big spaghetti-like strings in the cell. So that's mitosis. And just to further illustrate, because really by the time the DNA starts to condense, and really by the time the cell is going into synthesis phase, even here when the cell is in synthesis phase, it's basically, that's it. Its full attention is now divide. And it's not, it's not filtering the blood. It's, it's not acting as a hepatocyte. It's not acting as a renal tubular cell. Its whole focus, its whole purpose is, let's get this DNA replicated and let's get these puppies to divide. Because the DNA at this point it's busy replicating, and then of course during mitosis, when all of the chromosomes are condensed, they're actually not even available for protein synthesis or for transcription. It's not until the DNA has decondensed that now the various enzymes, the RNA polymerase and other enzymes, have access to the DNA. So we have this process of DNA condensation as mitosis begins, and then decondensation as the cells return back to the G1 phase, the interphase. Okay, meiosis is a different process where you start off with a cell that has 46 chromosomes, but you wind up with an egg or a sperm that only has 23 chromosomes. So what happens here in meiosis is first you have your chromosomes, but here you have not just 46 chromosomes, you really have 23 pairs of chromosomes. And so here, for instance, chromosome number 7 from mom and chromosome number 7 from dad are forming an intimate association during meiosis. Each of these chromosomes is going to replicate. So you have chromosome 7 from dad and you have chromosome 7 from mom. But in mitosis, half of the chromosome from mom and half of the chromosome from dad went into one daughter cell, and half of each of these went into the other daughter cell. But in meiosis, dad's chromosome goes one way and mom's chromosome goes the other way. And for chromosome 9, dad's chromosome goes one way and mom's chromosome goes the other way. And for each chromosome, whether dad's chromosome goes one way or the other way and whether mom's chromosome goes the other way, that decision for one chromosome is completely independent of the assortment of maternal and paternal chromosomes for the other chromosomes. And as a result, just based on this type of segregation, there's 2 to the 23rd possible combinations of possible gametes, or 8.4 million possible gametes. You know, this gamete might have chromosome 1 from dad, chromosome 2 from dad, chromosome 3 from mom, chromosome 4 from mom. This one over here might have chromosome 1 from dad, chromosome 2 from mom, chromosome 3 from mom, chromosome 4 from dad, etc. And if you look at all of the possibilities, you'll find that there's 2 to the 23rd or 8.4 million possible. So the chromosome for mom and dad line up. One goes one way, one goes the other way. And then there's a second cell division that takes place where now, just like in mitosis, these chromatids split and you wind up with the 4 gametes. And these gametes each have 23 chromosomes, or what are called, they're now haploid, whereas the original cell had 46 chromosomes, or 2 sets of the chromosomes. They're referred to as diploid. So a little bit of vocabulary. The DNA content in a normal cell is diploid, or 2 copies of every chromosome, whereas the amount of DNA in a sex cell is haploid, or only a single copy. These cells up here are referred to as body cells or somatic cells, as opposed to the gametes down here, the haploid cells. These are referred to as germ cells. So we have germ cells, which are haploid, and we have somatic cells, which are diploid. And I emphasize that a little bit because normally when we talk about genetics, we talk about mutations being passed from one generation to another. You have mutations here, and those are then transmitted through the sex cells. But when we start talking about cancer, it's different in that it has nothing whatsoever to do with meiosis. In cancer, you have mutations in somatic cells that are transmitted to daughter cells just during regular mitosis. So when we talk about meiosis, we're talking about somatic cells, diploid somatic cells leading to haploid germ cells, and that's the normal thing we think about when we talk about inheritance. But when we come back to cancer, we're going to be talking about diploid somatic cells through a process of mitosis yielding daughter diploid somatic cells. But if there's mutations that have arisen in those body cells through some process like radiation, those mutations then spread to the daughter cells as a mechanism, for instance, of cancer origination. So another way of looking at meiosis is we have the pairs line up, then the maternal goes one way, the paternal goes the other way, and then there's a second phase where the sister chromatids split. And it's also kind of interesting when you think about when this occurs. Meiosis 1 in females doesn't proceed all the way through until ovulation, and then meiosis 2 doesn't complete until fertilization. So you have a lot of oocytes or a lot of eggs that are sitting there for a very long period of time, not completing their separation until ovulation. Whereas in males, the process of meiosis 1 and meiosis 2 is continual, it's ongoing. And we'll come back to this later, but it's believed that this is the reason why diseases resulting in abnormal chromosome numbers, like trisomy, like Down syndrome, are most often due to non-disjunction in the female, whereas mutations in genes are more commonly due to mutations coming from the father's side. And so non-disjunction events usually come from the mother, and actual mutations usually come from the father. And that's because non-disjunction occurs because of the delayed progression of meiosis in the female, whereas the mutations are occurring in the male because you have so many mitoses and meioses occurring continuously throughout the life of the male. And then finally, for this particular portion, the cell division, we need to talk a little bit about crossing over. So here you have a dad's chromosome 7, and here you have mom's chromosome 7. And during the process of meiosis, one is going to go one way and one is going to go the other way. And that's going to lead to 8.4 million possible gametes. But before these two say goodbye, they shake hands. And of course, they just hate to part ways, and so they have to take a part of each other with them. Okay? And so there's a process of crossing over where these two chromosomes, the chromosome 7 from dad and the chromosome 7 from mom, literally exchange genetic material back and forth. And so what winds up happening is the chromosome that goes to the left in this sperm cell, or the chromosome that goes to the right in this other sperm cell, is not a pure copy of the chromosome they got from mom or the chromosome they got from dad, but rather it's a copy that's a little bit of a blend. And crossing over is really the rule. It's not the exception, it's the rule. There's an average of 3 crossing overs per chromosome. So crossing over is always happening. And so, you could say on the one hand, there's 8.4 million possible gametes, but really the number has got to be at least an order or two of magnitude higher than that, because crossing over can occur at so many different places. There are hot spots for crossing over, but we know that crossing over occurs really along the length of the chromosome. And so you might have one cell that crossed over like this, but another cell will have crossed over completely different. So the number of possible gametes that are being produced, or if you look at it another way, if you look at all the sperm that's produced in a male over their lifetime, you're not talking about 8.4 million different types of DNA. You're talking about billions, if not trillions, of different types of DNA, because the crossing over that takes place is different each time. And that's going to be very important. That crossing over tends to be important, number one, because it helps us, as we'll talk about later, figure out where genes are located. But the other thing that's important is crossing over sometimes introduces pretty interesting errors, and as a result, results in genetic disease. And so we'll need to understand a little bit more about crossing over, and we'll need to understand a little bit more about how crossing over is identified from a DNA testing perspective, and how mutations, crossover-based mutations, are communicated in HL7 standards. So we're going to switch now to the other slide deck on the central dogma of DNA. But before we do, let me just ask you guys a few questions. Somatic cells, are they haploid or diploid?
Somatic.
Diploid.
Diploid.
What's that? Diploid.
Diploid.
Right. Very good. Can anybody name an organelle participating in protein synthesis?
Ribosomes.
The ribosomes. Great. That's right. Okay. During what cell cycle phase does DNA replication occur? I'll give you a hint. If you down,
Mitosis.
so okay. So during what cell cycle phase does DNA replication occur? And the answer is it's the S phase, the synthesis phase. So what's happening here is the DNA is replicating way back here, and then we get ready. The cell plumps up. All the organelles have divided. The cell is nice and big and plump and ready. Then when you pass the checkpoint that says everything looks okay, then we go through mitosis. But the actual DNA gets replicated back here during the synthesis phase, or the S phase. And by the way, the answers are down here. So anyone who wants to cheat and get the slide deck, you know the answers.
Hey, Bob, real quick question. Do they understand how those checkpoints determine that everything is ready? And that's mind-boggling by itself.
I guess I would say to some degree. Everything is to some degree, and nothing's complete. And I don't know if I can answer much more thorough than that. It's, if you have mutations in certain proteins in a particular cancer, where the checkpoint goes out of whack. And we'll talk a lot more about this in the cancer discussion, but certainly like P53 has been an extremely well-studied protein that's responsible for playing a role in the second checkpoint and determining, hey, if things aren't looking good, send this cell to the recycler. P53 plays a role that's pretty well understood in, hey, you didn't pass, this cell needs to undergo programmed cell death or apoptosis. I think, when you consider that there's hundreds of proteins involved in the cell cycle machinery, then I would say, there's a handful that are understood pretty well, but there's a boatload that's not really understood well at all. If that helps, Ron.
Yeah, we're just scratching the surface on this particular piece as well.
So what I'm going to do now is switch to deck 3, which you don't have access yet to here because we're only going through the first half. Next week, we'll go through the second half of this deck, and we'll go through deck 4. So next week, you'll have access to deck 3 and deck 4. And we're going to just kind of sort of quickly go through the central dogma of biology. It's only going to take us about 10 minutes or so to go through. And I think, based on your responses, just for fun, I put this in red because it's, if you have any takeaways from today, take this away. But I think everyone already knows the central dogma of biology is the DNA is transcribed into RNA, and RNA is translated into protein. And it's obviously critical to understand genetics that the central dogma is understood. Not to say that there aren't now a growing list of central dogma variants. And next week, we're going to talk about the variants on the central dogma theme. But it still helps to really understand the central dogma. So here's DNA. The portion of DNA that's relevant for a particular gene is transcribed into messenger RNA. The messenger RNA is a faithful copy of a portion of DNA corresponding to a gene. The messenger RNA leaves the cell and enters the cytoplasm, leaves the nucleus and enters the cytoplasm of the cell. And ribosomes in the cytoplasm of the cell, ribosomes traverse along the messenger RNA, reading the messenger RNA and directing it to construct proteins or translate the RNA and construct a protein. So we're going to look a little bit more at the biology of the central dogma. Not a whole heck of a lot, but we're going to look a little bit at it. First thing, I don't know that anyone here is unclear about what is a protein, but just so that we've stated it, a protein is one of the 4 macromolecules of life. The macromolecules are considered to be nucleic acids, carbohydrates, lipids, and proteins. And proteins themselves are polymers comprised of amino acid building blocks. And as you'll see, that's critical to understand how DNA directs the development of protein. And proteins serve a boatload of roles: enzymes, hormones, structural, too many to elicit here. We're also going to talk a little bit about RNA. Messenger RNA is the DNA transcript. So you have a messenger RNA that's a copy of a segment of DNA corresponding to a gene, and that messenger RNA leaves the nucleus into the cytoplasm where the ribosomes translate it into a protein. But there's at least dozens of other types of RNA. We mentioned at the beginning that we now know that there's a boatload of genes that are transcribed into RNA, but the RNA is not translated into protein. And some of that RNA is called transfer RNA. Some of that RNA is called ribosomal RNA. There are microRNAs. There are small nuclear RNAs. There are long non-coding RNAs. You'll see that the vocabulary for RNAs is a little bit confusing, reflecting the different stages of our understanding. And so it's not as though there's from a terminology perspective, there's not really a nice hierarchical description of the different types of RNA, but there's a number of different types of RNAs serving various roles in protein translation and in regulation. And in fact, there's a boatload of RNAs where we simply have no idea what those RNAs are doing. And there's estimates there's as many as 25,000 non-coding, non-protein coding genes. So there's thousands and thousands and thousands of different RNA molecules in the nucleus and in the cytoplasm, some of which we understand, most of which we have no idea at this point of what it's doing or if it's even doing anything. Maybe the nucleus is just so good at translating the DNA into RNA that it does it, and maybe the RNA then either finds a home or it doesn't. There's a lot of controversy around, is all of the transcribed RNA even functional to begin with? RNA is chemically similar to DNA, but it's only a single strand rather than a paired strand. And where DNA contains a T for thymine, RNA contains a U for uracil. A little bit more about what's a protein, because to a large extent, and not to a complete extent by any means, but to a large extent, the ultimate three-dimensional structure of a protein is implied by the amino acid sequence. So if I tell you that I have a protein that's asparagine followed by glycine, followed by phenylalanine, followed by glutamate, followed by glycine, etc., then there's computer algorithms out there now that can infer what the corresponding secondary structures and tertiary structures are. And then from there, there's other computer software that can begin to predict, given the structure of that protein, what's its function. So at least to some extent, seemingly to a large extent, but certainly not completely, both the three-dimensional structure and the function of a protein is determined by its primary sequence of amino acids. So the primary sequence of amino acids is what's known as the primary structure of a protein. The secondary structure of a protein is referred to different motifs within that protein, such as beta-pleated sheets and alpha helices. And then ultimately, you have a complete three-dimensional protein, and that's known as the tertiary structure. But like we saw with hemoglobin, multiple proteins that come from individual genes can glom together to form a quaternary structure, which is a complex of protein molecules. And this is, for instance, what we saw with hemoglobin, which is a tetramer of 4 protein molecules. Okay, so again, the messenger RNA comes out of the nucleus, and these ribosomes, these little blue guys here are the ribosomes, and the ribosomes just kind of ride along the messenger RNA, reading it, and the proteins just kind of grow. So actually, this ribosome is moving this way, and so you have the ribosome here, and it moves here, and as it's moved, the protein chain has gotten longer. The ribosome moves to here, and the protein chain gets longer. The ribosome moves to here. And for very highly active metabolic cells, like if you're talking about a red blood cell that's still in the bone marrow, its whole purpose in life is to produce hemoglobin, you'll see there could be literally hundreds or thousands of hemoglobin messenger RNAs in the cytoplasm, and each messenger RNA could have dozens or hundreds of ribosomes riding along it, so that you're producing thousands of copies of hemoglobin protein during the development of that red blood cell. So the question is, what is this ribosome actually reading on this DNA, on this RNA, and how is it actually then determining how to grow this protein, what amino acid to put next in this growing chain of protein? And here's really where the central dogma comes into play. So we have the ribosome here, and along the ribosome we have the bases. Just like in DNA, we have the bases. And what happens is every 3 bases, every unique combination of 3 bases corresponds to a specific amino acid. So here, the ribosome, the messenger RNA has an AAA, and that corresponds to an amino acid of lysine. The next set of 3 bases in the messenger RNA would be a GAU, and that's going to tell the ribosome, okay, connect, and asparagine. The next set of 3 bases on the messenger RNA is a UUC. That's going to tell the ribosome, okay, now let's connect a phenylalanine. So the ribosome reads 3 bases at a time. Those 3 bases are called a codon, C-O-D-O-N, a codon. A codon is a sequence of 3 bases that's read by the ribosome, and every unique set of 3 bases corresponds to a specific amino acid. And so when the ribosome reads these 3 bases, it knows exactly what amino acid to tack on next to the protein. Notice, however, that the notion of frame is critically important, right? Because if the ribosome, rather than reading AAA, was reading GAA, you get a fundamentally different protein. And so the messenger RNA has a very explicit start, okay? Because the reading frame has to be explicit in order to ensure that you get the exact correct protein, okay? So there's an explicit start, and from that explicit start, then every count of 3 bases determines what the next amino acid is in the chain. Okay, so this is very important, because that's how you begin to understand that if there's a mutation in this A, if this A, for instance, were a G, then rather than the ribosome sticking a lysine in here, it's going to stick in a different amino acid, whichever amino acid happens to correspond to AGA. Is there anyone that's fuzzy on that? Because this is super important, and we'll talk offline or come back to this more next week if that's not really clear to people. Okay, and again, if you're embarrassed to say something, send me a note, so we can go over this a little bit more before the next class. So the ribosome is reading 3 bases at a time, and we have this thing here called the genetic code. So depending on what the 3 bases are, you can figure out exactly what the corresponding amino acid is. So if the codon is a CUU, then the corresponding amino acid is going to be a leucine. If the codon is an AAA, the corresponding amino acid is going to be a lysine. If the codon is a UGA, it's going to tell the ribosome, terminate the protein, we're all done. It's called a stop codon. Okay? So for every possible combination of 3 bases, there is a corresponding amino acid that will be inserted next into the growing protein, or that particular sequence will be a stop codon. The AUG codon is critically important because that signals the start of the reading frame. So every single messenger RNA will include AUG, and every single protein in humans that we know of will initially begin its protein translation with MET, although in almost all cases the MET will be subsequently removed by post-translational modifications. So you can always look at a messenger RNA and see where the reading frame begins by finding the AUG. That means every protein synthesis will begin with a methionine, but the methionine will be removed. You won't see the methionine in the final protein. So just to review the central dogma, in the nucleus we have the 5' strand, okay, and what's actually being transcribed is the 3' strand, And when the 3' strand is transcribed, the RNA is complementary to the 3' strand, but similar to the 5' strand. So when we look at DNA sequences corresponding to genes, we'll typically look at the 5' strand, even though that's the non-transcribed strand. We'll look at that because it's the same as the RNA. The RNA, then you wind up getting the RNA, but again, the RNA initially contains the exons and the introns. So the initial transcription of RNA has all of the introns. The introns have to be spliced out. And so you wind up ultimately with a mature messenger RNA where all of the introns have been spliced out and a bunch of A's or a poly-A tail have been added for chemical stability. You now have a mature RNA that leaves the nucleus, and now ribosomes ride along the mature messenger RNA and generate the proteins. Ultimately, these proteins work their way into the Golgi apparatus, where they are further modified and sent about their way to do their business. We went through that very fast, but we have a few minutes left for a quiz. Okay? We have a few minutes left for a quiz. If UAU, which is right here, UAU is a codon that codes for a tyrosine. If UAU is mutated to a CAU, how will the amino acid change?
It would change from tyrosine to HIS, I guess, histidine.
To histidine, that's right. Okay. And what if UAU is mutated to a UCU?
Change to SCR.
It would change to a serine. To a serine. What if it's mutated to a UAC?
It would change to leucine.
No, tyrosine. It would remain as tyrosine.
Will remain the same.
Oh.
And actually what you'll see is the third base in a codon is often referred to as the wiggle base, because mutations in the third base of a codon generally don't change the corresponding amino acid. They result in what are referred to as synonymous mutations. And so here UAU changes to a UAC or UGU changes to a UGC, the amino acid doesn't change. What if UAU is mutated to a UAG?
It is a stop codon.
Right, and that can really be bad. Imagine if this mutation is early on in the protein chain, you basically just have produced a very tiny protein. What if a UAU has an insertion, if a C is inserted, and you now have a UACU?
Would it still be tyrosine?
Everything gets moved,
It would
doesn't
not,
it?
right? It would not be readable, right?
So you'd still have a tyrosine,
Right.
but you've just upset the reading frame.
Right.
Everything downstream, you've just changed all the codons for everything downstream. So if this mutation is early in the messenger RNA, you could produce a devastating effect on the protein. But if this mutation, for instance, is near the tail end of the messenger RNA, you may only have a minor effect on the protein. But the amino acid here doesn't change, but it's the fact that these insertions can change the reading frame that can make them potentially so devastating. Listen, do we have 2 more minutes? I want to do something real quick, if we can. again, this is a dry run class, and I am super indebted to you guys for coming, and I hope you're getting something out of it as well. I would love to get some feedback now or by email. What about this class did you like, and what about this class did you think could change? Maybe we can spend a minute or two before people drop off. And actually, I'm more interested in the change. I love to hear what was good, but I want to know if people have any constructive feedback about where the class could improve.
Bob,
Hey, Bob.
this is Ron. Yeah, I would say the only thing I would change would have been the amount of time that you spent demonstrating the various online sources for codifying all this stuff.
To increase it or decrease it?
Decrease it for this particular class.
Was it overwhelming, or was it just it steered away from the meat of the presentation?
I found myself tuning out a little bit, to be honest with you. It just seemed like it was just way too much material for this stage. For me, anyway. That's just my personal or time spent, I guess.
Anything else?
Just a general glossary of terms will be helpful.
By the way, for every deck, the specific terms we don't go back over them, but the specific terms are in the vocabulary. You'll see that there's a vocabulary slide in every deck.
Oh yeah. Okay. Thank you.
And in deck 1, we started you off with a big. If you go back to the course overview deck, we started you off with a nice little starter set. So I understand that there's hundreds of words here, and we'll try to make the vocabulary as painless as we can as well. By the way, every vocabulary word down here we will have covered during the class as well.
Sure. Thanks.
Anything else?
Well, I do like seeing the interspersing of the vocabulary and the standards.
Interspersing.
I don't know if anyone else does that. Maybe just be where I'm coming from, but I'd be interested in hearing from other folks if they felt that was out of place at this time, or they liked it. It's okay if you don't like it.
I'm not sure I follow what you were saying. Actually.
Well, I thought I heard you say that early that you didn't like the online sources and then the examples, or you thought maybe it was a little out of place. So, I'm curious to see when he started going into like the SNOMED code and the codes and the the message examples, whether that was good at this time or not.
Well, if you're referring to what I said, I thought that was right on. I mean, I do like dipping into both. I just was trying to say that it was a little bit, in my opinion, going to the various sites and showing, where you can search by genomic region and so forth and so on. It was interesting, but it was just a little bit too much time. That was all I was trying to say.
Yeah, and it's funny, for myself, I already have bookmarked all the sites that Bob mentioned, because I want to have them for reference. So
I agree, totally agree. I was just trying to say the amount of time. That was all.
Okay.
Right. Yeah. It's a bit overwhelming too, and I sometimes what I do is deliberately throw a lot at you so that over the weeks to come, when I say these words again, it's like you will have heard them once, and maybe sometimes that's not the best thing to do.
By the way, I like the quizzes, and I'm just so glad that I'm not actually working for a degree anymore. And I hope you don't. I hope you don't report any of my answers to the previous institutions which have agreed, because I don't want any of them taken away. Okay.
No. But homework is coming soon. Just as a heads up. So. all of a sudden after that session, for some strange reason, no one will be showing up to class anymore. But, all right. Well, thanks again, everybody. Really appreciate your time, and any other feedback, I would love to see it. Thanks, everybody.
Thanks, Bob.
Thank you very much.
Thank you very much, Bob.
And very well presented. Thank you.
I agree, totally.
Bye.
Thanks.
Thank you.